fellow lab rats, this is Rebecca from the Lab Rat YouTube channel. In this video, we're going to be discussing the Rh blood system and blood banking. All right, let's get started. The Rh blood group system is extremely important in blood bank. It is second only to the ABO blood group system in importance in transfusion medicine. It's a complex system containing over 50 different Rh antigens. Antigens within the Rh system are very immunogenic. And when we're discussing immunogenicity, this is referring to the ability of an antigen to stimulate an antibody response. So if a patient does not have a specific antigen, and we're talking about antigens that are present on the red cells in blood bank, okay? So if they do not have a specific antigen on their red cells, and that patient gets exposed to that specific antigen from either a blood transfusion or a pregnancy, the body can develop a specific antibody targeted to that foreign antigen. So if you are transfused or are pregnant and get exposed to the fetus's blood, and those red cells contain antigens that your body does not have on their red cells, the body goes, oh gosh, what is that? That's a foreign invader. That's a foreign invader. We're going to launch this immune reaction and develop antibodies targeted to that specific foreign antigen. Okay, so that's what we say when these antigens within the Rh system are very immunogenic. So they cause antibody production, and you know that antibody is stimulated to be produced very frequently. So recall that in the ABO system, the antibodies within that system are naturally occurring. So for example, a patient with A blood, right, just naturally has anti-B antibodies present. So again, this is not the case for the Rh system. Patients again have to be exposed to the Rh antigen through transfusion or pregnancy in order for antibodies to develop. Rh antigens reside on proteins, which is different from those in the ABO system, which are on sugars. So these are associated with the membrane of the red blood cell, most specifically with the integrity of the membrane. So again, Rh antibodies are stimulated from transfusion or pregnancy, and they can be implicated in transfusion reactions and in something we call hemolytic disease of the newborn. So if a patient is Rh negative and is transfused with Rh positive blood, they develop an antibody towards that foreign Rh antigen that they receive from the transfusion, and that antibody hangs out in the bloodstream. And let's say a couple months later, they're transfused again with Rh positive blood again. What will happen there? So that Rh antibody in the patient that developed from the previous exposure of that foreign Rh antigen will essentially attack. So it'll glutinate with the foreign red cells from the new transfusion, and this is referred to as a transfusion reaction. Hemolytic disease of the newborn occurs when a mother and fetus's blood types are incompatible. So if a mother has an Rh antibody from a prior transfusion or even a prior pregnancy, and the fetus she is currently pregnant with is Rh positive, that Rh specific antibody can go and attack that Rh antigen on the blood cells of the fetus, which causes a destruction of that fetus's red blood cells. So this is hemolytic disease of the fetus and newborn. Rh positive and Rh negative are used when referring to a patient's blood type. So for example, O positive or O negative, A positive, A negative, AB negative, AB positive, those kinds of things. So that Rh positive indicates that a patient has the D antigen on their red blood cells. Rh negative indicates that a patient does not have the D antigen on their red blood cells. So before 1939, we only knew about the antigens of the ABO blood group system. Blood transfusions were only based on this ABO system. So for example, if somebody was determined to have A blood, they gave them A blood. Somebody had B blood, they gave them B blood. But there were still deaths involved with transfusions. So Levine and Stetson had a hemolytic transfusion reaction occur in a pregnant patient. The mother delivered a stillborn fetus and required blood transfusions. So this mother's husband had the same ABO blood type as the mother, and so his blood was used for the transfusion. So his blood was transfused into his wife's body. After the transfusion was performed, the mother/the wife had an acute hemolytic transfusion reaction. So these scientists were able to isolate an antibody from that mother's serum. Levine and Stetson postulated that the fetus and the fetus's father had a common factor in their blood that the mother didn't have. The mother was pregnant with the fetus and produced an antibody when exposed to that foreign antigen from the fetus that was, of course, present on the fetus's red cells. Landstein and Weiner reported that they found an antibody that was developed by guinea pigs and rabbits when they were transfused with rhesus macaque monkey blood. So this antibody agglutinated or reacted with 85% of random human red blood cells that were tested against it. So this antibody was called Rh after the rhesus monkey. This had the same pattern of reactivity as the antibody from the mother described by Levine and Stetson. So now, many years later, it was determined that these two antibodies were different. However, the name stuck. The human produced antibody that was discovered first in the obstetric patient is referred to as Rh, and the antibody produced by the guinea pigs and rabbits is renamed to anti-LW or antibody LW in honor of Landsteiner and Weiner. At the beginning of this lecture, I stated that there were over 50 antigens within the Rh system. That is true. However, there are only a couple that are the most common and super important for us in blood bank. Big C and little c. Big E and little e are antithetical genes. Antithetical is a term used when describing a pair of antigens that are coded by different alleles of a single gene. So think back to our Punnett squares, and we learned about Punnett squares in our blood bank genetics lecture. If you have not seen that lecture, stop this video now and go and check out that lecture before you continue. So think back to the Punnett squares example. So I'm going to use the C gene as an example. So a patient can either be big C/big C, or big C/little c, or little c/little c. Each of those C types is inherited from one parent. So, for example, a big C/little c patient would have inherited a big C from one parent, and the other parent gave them the little c. It is often assumed that little d would be the alternate allele of big D, but that's not the case. Little d isn't an allele. It is just used to indicate the absence of big D. So this is a chart that shows the gene frequency of big D, absence of big D, denoted as little d, big C, big E, little c, and little e in the United States. So I know there are many viewers of my channel that are outside of the United States. Shout out to you. Thank you for supporting my channel. I am based in the United States, and my medical lab students are here as well. So if you're from outside of this country, your gene frequencies may differ from these. For those of you who are in the United States, you need to memorize these frequencies. You likely will be asked to list these in order of highest to lowest frequency as an example, or perhaps given a couple of these antigens and asked which one has the highest frequency or the lowest frequency out of the group. So make sure you know this chart. Terminologies used to describe the Rh blood group system are derived from 4 different theories. 2 of them, Fisher-Race and Weiner, are postulated from genetic theories of how Rh is inherited. Rosenfield et al. Is used to describe either the presence or absence of a given antigen. And the 4th is the result from the collaboration of the International Society of Blood Transfusion, or ISBT. First, let's talk about the Fisher-Race theory. So in the 1940s, Fisher and Race postulated that Rh antigens were determined by 3 closely linked genes that code for 3 pairs of alleles. These were inherited as a unit, or also called a haplotype, and each person inherits a set of Rh genes from each parent. Fisher and Race named the antigens big C and little c, big E and little e, and big D and little d. Now remember, little d doesn't actually exist. It just represents that the big D antigen is not present, of course, on the red cells. So this theory ended up actually being too simplistic to account for the entire Rh system, but is still used in current blood making to determine Rh phenotypes. In the Fisher-Race terminology, there are 8 possible combinations using 6 gene haplotypes. So each of these 3-letter combinations is a haplotype, and each haplotype is inherited from each parent. So the haplotypes are big D, little c, little e, big D, big C, little e, big D, little c, big E, big D, big C, big E, little d, little c, little e, little d, big C, little e, little d, little c, big E, and little d, big C, big E. Hopefully I said all of those correctly. It's kind of like a tongue twister, but they are written here correctly on this PowerPoint. Because the Rh genes are codominant, each inherited gene expresses its corresponding antigen on the red blood cell. In rare occurrences, a patient may fail to express big C or little c, big E or little e, or all the big C, little big E, little e antigens. An Rh positive patient that fails to express the big C or little c antigen is written as dash big D, big E, or dash big C, big D, little e. A patient that has no expression of Rh antigens is called Rh null, and the phenotype is written as dash 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 dash. This chart shows the 8 possible Rh haplotypes and their prevalence in the United States in the white, black, and Asian populations. As you can see, the most frequent Rh haplotype in both Asian and white populations in the United States is big D, big C, little e. The most frequent Rh haplotype in the black population in the United States is big D, little c, little e. The next Rh terminology theory is the Winer-Rh-HR terminology. Winer believed in his studies that there was only one gene responsible for defining a patient's Rh status. This gene produced something he called an agglutinogen that contained at least 3 factors. The agglutinogen can be considered the phenotypic expression of the Rh haplotypes. So this chart on the right-hand side here shows the major agglutinogens and their factors. It also shows their shorthand designations and their corresponding Fisher-Race antigens. In this chart, a capital R indicates the presence of big D, little r indicates the absence of big D, prime or 1 indicates the presence of big C, double prime or 2 indicates the presence of big E, Z or Y indicates the presence of big C and big E. Winer terminology is complex, as you can see here. So blood bankers use a modified Winer terminology. What you need to memorize from this chart are the columns that are labeled with an asterisk. So the far right 2 columns here. So you need to know which Winer shorthand designation corresponds to the Fisher-Race terminology. So for example, a patient who is little r/little r prime would be little d, little c, little e / little d, big C, little e in the Fisher-Race terminology. In the 1960s, Rosenfield and their associates proposed a system that assigned a specific number to each antigen. They were assigned in order of their discovery or relationship to the Rh system. This system has no genetic basis to it. So this is Rosenfield nomenclature. So Rh1 represents big D, Rh2 is big C, Rh3 is big E, Rh4 is little c, and Rh5 is little e. And yes, you do have to have these memorized. Okay, so let's do some practice here involving the conversion of most probable genotypes between Fisher-Race, Winer, and Rosenfield. Okay, so this is the first practice problem. So a patient has the Winer genotype of R1, so big R1, little r. So what is this patient's most probable Fisher-Race genotype? Okay, so a few slides ago, I had a big chart here for you, and I said that the important things that you need to remember on that chart are the 2 columns that have the little asterisk. So what I've done is basically cut this chart apart and just given you those 2, excuse me, those 2 ones that have asterisks. So on the left-hand side column, we have the shorthand designation, and then we have So the shorthand designation is the Winer type, and then the Fisher race antigens. Okay, so this Winer genotype is big R1, little r. Okay, so this means that R1, so big R1, came from one parent, and the little r came from the other parent. So we need to think of it as big R1 slash little r. Okay, so all you got to do is look up R1. Let me see here. So R1 is here. So this is the first haplotype, okay? And then the little r here. This is the second Fisher race haplotype. Let me get off this. Okay, there we go. So big R1 is equivalent to big D, big C, little e in Fisher race. Okay. Little r is equivalent to little d, little c, little e in Fisher race. So you just combine them. So this patient's Fisher race genotype is big D, big C, little e, slash little d, little c, little e. That's all you have to do, okay? Now the same patient, okay, the modified Winer genotype of big R1, little r, and then we determine that the Fisher race genotype is big D, big C, little e, slash little d, little c, little e. What is the Rosenfield nomenclature of this patient? So again, you need to memorize the Rosenfield designations, okay? And I've listed them for you here on the left. So RH1 is equivalent to big D, RH2 is big C, RH3 is big E, RH4 is little c, and RH5 is little e. So we just need to look at this Fisher race genotype and plug it in. So here we are, big D, big C, little e, slash little d, little c, little e. So first one here, we're just going to go down the groups of them there, okay? So big D is 1, okay? Big C is 2, okay? And so I'm comparing them to here, okay? Little e is what? It's 5, right? So here's 5. And I'm getting that from here. So I'm just doing this one first, then this one, then this one, and then just continue on, okay? So little e is 5, okay? Then the next one in the group is that little d, right? There's nothing for that little d. We're just going to leave that one blank. And then we're going to go to that little c, which is 4, okay? And then, of course, we've already done little e from that first haplotype, so it's just 5. So we have 1, 2, 5, and 4. We don't have 3, right? Because there's no big E present. So the Rosenfield phenotype is 1, 2. We don't have a 3 present, so we're going to do -3 or -3, 4, and 5. So this is the Rosenfield type. That's how we convert them. Okay, let's do another one. So a patient has the modified Winer genotype of big R1, big R2. What is this patient's Fisher race genotype? So again, big R1, big R2 means big R1 came from one parent, and that big R2 came from the other parent. So think of it as big R1 slash big R2. Okay? So let's go to R1. Okay, that's big D, big C, little e, and then R2 is big D, little c, big E. Okay, so R1, big D, big C, little e, R2, big D, little c, big E. We're just going to put them together, right? So each haplotype. So this patient's Fisher race, most probable Fisher race genotype is big D, big C, little e, slash big D, little c, big E. Okay, so that's your answer. Okay, same patient. So the Winer genotype of big R1, big R2. The Fisher race genotype is big D, big C, little e, slash big D, little c, big E. Okay, so what is the Rosenfield nomenclature of this patient? So again, I have on the left-hand side the Rosenfield designations, and you do need to memorize those, right? Same with the other designations. You need to have these all memorized, okay? So here is the Fisher-Race genotype, and again, we're going to do this one first, this one, this one, this one, this one, and this one, and we're going to compare it to that Rosenfield nomenclature to get our result. So big D is the first one. That is 1. Next one is what? Big C. That's 2. Little e is 5. Then the next one is big D again, right? So we don't have to list it again. Then little c, okay? Little c is, we're right here in this practice problem, okay? Little c is 4. And then the next one is big E, which is 3. So we have 1, 2, 3, 4, and 5. We have all of them this time, so we don't have to do any dashes. So the Rosenfield phenotype is Rh 1, 2, 3, 4, and 5, okay? So why would we have, dash 5? Well, we would have dash 5 as an example if we didn't have little e present. We would have dash 4 if we didn't have little c present. We would have dash 3 if we didn't have big E present. We would have dash 2 if we didn't have big C present. So that's where those dashes come in. And there's no dashes here because we have all of the possibilities. The International Society of Blood Transfusion, or ISBT, developed a uniform nomenclature that is able to be read visually and also be read by machines. So this is a 6-digit number for each antigen belonging to a blood group system. The first 3 of the 6 numbers refer to the blood systems, and the following 3 numbers correspond to the antigenic specificity. The results of Rh antigen typing do not define the genotype, only the phenotype. So remember, in our blood bank genetics lecture, again, if you have not watched that yet, you really need to stop that and watch it before you continue with this video. So in that lecture, remember the black pugs and the fawn pugs that we did Punnett squares with, right? So the color of the pug is the phenotype, so whether it's black or fawn. So the genes that make the pug either black or fawn, that is the genotype. So when we type a patient for their Rh antigen status, we only know their phenotype, which is Rh positive or Rh negative. The most probable genotype correlates Rh phenotypes to the most probable genotypes based upon the patient's race and frequencies of occurrences in the race. So here's a most probable genotype example. So here's the testing on a patient. So we have 0 under anti-D, anti-big C, and anti-big E. So this means that there was no reaction between the patient red cells and the corresponding antibody. So the corresponding antibody is either big D, big C, or big E reagent. So let me just write on here. Let's use purple. Why not? Okay, so patient cells here. Okay, patient cells reacting with anti-D antisera. Okay, this is a reagent that has antibody towards big D. Then it's mixed with a reagent that includes the antibody for big C. This one is a reagent that includes an antibody for little c. And this one includes a reagent this reagent includes an antibody for big E, and then lastly, this reagent includes an antibody for little e. So what I'm talking about here is this result, and this result. There's 0 there, okay? So that means that there is a negative reaction, which means that the patient is negative for big D, big C, and big E. So big D, big C, and big E, okay? So these are considered negative reactions. Now there's a 4+ under anti-little c and anti-little e, which is, let's do green for these. 4+ here and a 4+ here. So that means little c positive and little e positive. So that means that the patient has a little c antigen and a little e antigen present within their red blood cells. And the reason for that is we're mixing the patient's red cells, okay, with reagent that includes those antibodies. So if we're having a reaction and we're going to get an agglutination in this part here, it's because it contains the corresponding antigen for that whatever antibody is present in the reagent. So in this particular case, We know that they have negative reactions and big D, big C, and big E, and positive reactions with little c and little e. So we know they're little c positive, little e positive, and negative for all the other antigens. Okay, so we're going to take the same person, okay, the same patient, and figure out the most probable Fisher-Race terminology. So again, the patient is big D negative, big C negative, little c positive, big E negative, and little e positive. So we know that because there is no big D, that means we must have little d's, which again represent the absence of big D. So there we go. So we know that there is no big D antigen, okay? So the first portion of our Fisher-Race genotype is going to be little d dash dash slash little d dash dash. So let's stop here, and I'm just going to re-explain this, okay? I don't want to confuse any of you. So we know there's no D antigen. How do we know that, right? Because there was a negative reaction here, okay? So big D negative, right? So what's left if we don't have a big D, right? Little d means that the patient does not have big D present, okay? So the only outcome for that, the only possibility is that they have two little d's, okay? Let's go to this next one here, okay? So we have no big C antigens, only little c antigens. And again, how did we know that? Let's use a different color, right? Big C is negative here, okay? We have no big C antigens. We only have little c antigens, and we figured that out right here, okay? Remember, little c is positive, big C is negative. So our next portion of the Fisher-Race genotype is going to be little c, right? So it's little d, little c dash slash little d, little c dash, okay? All right, last part. We're going to look at E's, right? So again, let's use yellow this time. We have no big E antigen, and we determine that because right up here, right, remember it did not react, the patient's cell did not react with that anti-big E antibody reagent, okay, that big E antisera. Excuse me. So we have no big E antigen. We only have little e antigens because remember right here is positive, right? So the third portion of our Fisher-Race genotype is we're going to have little e's, right? So we have little d, little c, little e, slash little d, little c, little e, okay? Okay, so let's go to the next portion of this, okay? So we're going to convert it to a modified Wiener terminology, okay? So again, our phenotype is this. Let's use purple here. Big D negative, big C negative, little c positive, big E negative, and little e positive. And again, on that last page, we determined that the Fisher-Race designation is little d, little c, little e, slash little d, little c, little e. Okay, and we figured that out based on these results here. Okay, so what do we have to do to determine the Wiener, the modified Wiener designation? So again, here is this beautiful chart right here. You need to have this memorized. Okay, so we're just going to look at it. Where's little d, little c, little e here? There we go. Okay, little d, little c, little e. What's the corresponding modified Wiener designation? Little r. Okay, so we have two little d, little c, little e's, so it's going to be little r, little r. Okay, so the modified Wiener is going to be little r, little r. Now that we've talked about the Rh terminologies, and of course 2 of those were derived from theories of genetic inheritance, we can now talk about the inheritance of Rh. So we now know that the inheritance of Rh is controlled by 2 closely linked genes present on chromosome 1. These 2 genes are called RhD, which codes for the presence or absence of the RhD protein, and RhCE, which encodes for the presence of Rh big C little e, RH little c big E, RH little c little e, or RH big C big E proteins. There is another gene called Rh-associated glycoprotein, or RhAG, and this is present on chromosome 6. So this gene helps to form complexes with the Rh proteins. If any mutations in this gene occur, it can cause either missing or altered RhD and RhCE proteins. It's also important to know that Rh antigens are inherited as codominant alleles. So recall what codominant means from our blood bank genetics lecture. So it means that neither gene in a pair of genes are dominant over the other. They are just both expressed. As we have already discussed when we were talking about Fisher race, offspring inherit an Rh haplotype from each parent. So a combination of DCE, so those 3 letters are one haplotype, whether or not they're capital C or capital E or little c or whatever. I mean, that just those 3 letters is considered a haplotype. So crossing over is a rare event because Rh genes are so closely together on the chromosome. So crossing over happens during meiosis when chromosomes of the same type are lined up. So sometimes parts of the chromosomes can be switched, so this is called crossing over. So this is rare with Rh because the Rh genes are so close together on the chromosome. So to explain the point of how these haplotypes are inherited, here's our friend the Punnett square. So again, if you need Punnett square practice, go back and check out the blood bank genetics lecture. Okay, so this is just an example, okay? We're going to figure out the percentages of offspring from crossing two parents and their haplotypes. So the first parent is big D, big C, little e, / little d, little c, little e. So that's one parent. And then they're going to produce offspring with a parent that is big D, little c, big D, little c, big E, / little d, little c, little e. Okay, so we're going to put one parent here on the left-hand side, and then we're going to put another parent's haplotypes up, their 2 haplotypes up on the top here. Okay, so we're going to assign the haplotypes of this parent on the left and assign the haplotypes or distribute the haplotypes of the parent up on the top. Okay, so this first square here, this upper left-hand square, so we should be able to do these Punnett squares in our sleep now, right? So we're going to combine it. So it's going to be big D, big C, little e, / big D, little c, big E. Okay, so this one goes here, okay? This one goes here, right? And we're going to do the same thing for all of these. So bottom left-hand corner, little d, little c, little e, / big D, little c, big E. Okay, and again, this one goes here, this one goes here, this one is coming from there, right? Okay, again, top right corner, big D, big C, little e, / little d, little c, little e. And then again, little d, little c, little e, and little d, little c, little e, okay? So here are the percentages. So 25% chance of all of these different inheritance. So again, 2 haplotypes here. This is a haplotype, and this is a haplotype. This is a haplotype, this is a haplotype, right? This is how they are inherited. So how do we test for Rh in the blood bank laboratory? The testing of D is included in what we call the forward grouping or the front type in ABO testing. I've already discussed how to perform this in my ABO blood group system lecture, but this bottle on the left-hand side here of the screen labeled anti-D is used. So this reagent contains antibodies toward big D. So 1 drop of this reagent is placed in a tube along with 1 drop of a patient's red blood cell suspension. It's mixed and placed in a centrifuge and spun according to the manufacturer's recommendation. So of course follow your laboratory's procedures please on this, but it's usually around 15 seconds in the centrifuge. When read, after this immediate spin step, if there is agglutination, this is considered Rh positive, meaning the patient has the big D antigen present on their red cells. If there's no agglutination, this is considered Rh negative, meaning the patient does not have the big D antigen present on their red blood cells. Most patients that are Rhd positive react with that anti-D antisera reagent in that immediate spin phase. So when I say immediate spin, I'm referring to putting the patient red cell suspension in with the anti-D Antisera and spinning it immediately in the centrifuge. So the results are read directly after the tube is done spinning. So most RHD positive patients are positive for this. Some patients have a weak D positive antigen expression. A weak D test, it's actually formally known as a DU test, needs to be performed on these patients to determine their RHD status. So I'm going to be discussing the different mechanisms of weak D, and then I will talk about how the testing is performed. So there are 4 mechanisms for the weak D phenomenon. So genetic weak D, the position effect, D mosaic or partial D, and DEL. Genetic weak D occurs when a gene that codes for a weakened expression of the D antigen is inherited. This is due to mutations. In this case, D antigens are complete, but they just are present in fewer amounts on the red blood cells. Genetic weak D is most frequently found in the Black population in the United States and is often associated with the R0 gene. This is rare in Caucasians, excuse me, but when in Caucasians, it's associated with the big R1, big R2 genotype. Individuals with genetic weak D rarely make anti-D antibodies. The position effect is when one gene interacts with another to modify and weaken the D antigen expression. This is seen most often when the big D gene is accompanied by the big C, little d, little e haplotype. When the big C gene is trans, meaning on the opposite chromosome to the big D gene, the steric arrangement of the big C antigen in relationship to the big D antigen appears to interfere with the expression of big D. It's not possible to distinguish between a genetic weak D from a positional weak D. Family studies are required if indicated. D mosaic or partial D is when the gene may not code for the total big D antigen. It's proposed that the big D antigen is composed of several antigenic subparts and they can be absent in rare occurrences. So this is attributed to hybrid genes with RHD being replaced with RHCE. Patients with D mosaic or partial D can be sensitized to the missing epitopes and produce anti-D antibodies, which can cause hemolytic transfusion reactions and hemolytic disease of the newborn. DEL is a phenomenon that occurs in patients whose red blood cells have an extremely low number of D antigenic sites on them. Most anti-D Antisera reagents are unable to detect it. Adsorbing and eluting the anti-D is the only way to potentially detect the antigen. So this DEL is most common in the Asian population, accounting for around 10 to 30%. It's very rare in the Caucasian population. So I use the term adsorbing and eluting. So adsorbing in blood bank is binding antibodies to red cells in an effort to remove them from a patient's plasma sample to better analyze antibodies that may remain behind. Elution is the process of removing an antibody that is attached to the surface of the red blood cell, allowing it to be free and usable for identification in the lab. So weak D testing, when is it required? So it needs to be performed on all donor red blood cells that type as Rh negative. It's not a requirement for the recipient of red blood cells. So why is this? So pause this video and think about it for a minute here. So why is this the case? Why does donor Rh negative blood need to be weak D tested? Okay, seriously, pause the video, think about it. Why does donor Rh negative blood need to be weak D tested? Okay, I'm holding you to it. You better have paused it and thought about it. Okay, so let's say we have a donor unit that is appearing as typing as O negative. The unit of red blood cells will be labeled as O negative, right? It will be given to other Rh negative patients. And that, in theory, will be compatible, right? Rh negative given to Rh negative, right? So what happens if this O negative donor had a weak D? It's going to type originally as Rh negative, but it has a weak D. And remember, weak Ds can cause antibodies to develop. So let's say we didn't know this donor red cell unit was weak D positive, and we gave this O negative weak D positive unit to a patient that had an anti-D antibody. What would happen? Likely a transfusion reaction, right? So let's say, it's another example here. Let's say we gave this O negative weak D positive red blood cell unit to an O negative patient. This patient would likely develop an anti-D antibody in response to being exposed to that foreign D antigen. So this isn't a good situation, right? Excuse me. So all Rh negative donor units need to be weak D tested to ensure that they actually are Rh negative. And when I say all Rh negative donor units, I'm talking about red cell units, okay? So they need to be ensured that they are actually Rh negative. Another scenario where weak D tests need to be run is when Rh negative mothers deliver babies. So they should be weak D tested to determine their candidacy for something called Rh immune globulin, or RhoGAM. So we'll talk more about RhoGAM in a couple of slides here, but basically if a mother is Rh negative and the baby that she delivers is Rh positive, if she is exposed to the baby's blood during the delivery, she could develop an anti-D antibody towards the baby and future pregnancies. So we give something called RhoGAM to Rh negative mothers, and this prevents an anti-D antibody from being made. So the mom will need weak D tested at delivery. Only Rh negative patients can develop an anti-D. Weak D testing uses the same reagent that we use for regular D typing at first. So the little bottle here labeled anti-D on the left-hand side of the screen here is the anti-D Antisera reagent. So you want to drop 1 drop of this and 1 drop of the patient's red blood cell suspension in a tube. Mix them and then centrifuge according to the manufacturer's specification. It's usually 15 seconds in the centrifuge. Then read after centrifugation. If there is agglutination, then the patient is labeled as Rh positive. If it's negative and you're performing a weak D test, then you want to mix the tube again and incubate at 37 degrees Celsius for 15 minutes. After that 15-minute incubation, spin again in the centrifuge. Read again and record results. Then, after that step, wash the cells and the tube 3 times, either manually or with an automated cell washer. After the washing steps, add 1 drop of anti-human globulin, or AHG reagent, in the tube and spin again. If no agglutination, record as negative and then add Coombs control to ensure that the AHG reagent is properly working. If there is agglutination, record as positive. Now, if you're one of my medical lab students, we will be performing weak D tests in the laboratory, and you will have a written procedure that walks you through step by step how to perform this procedure. So don't get too stressed out about it here in this presentation. I just know as a blood bank student myself, I would go over the PowerPoints and had a difficult time relating the PowerPoint material to what we were actually doing in the lab. So I wanted to include a summary of the procedure to try to make things easier to understand and correlate. So I also do have a weak D procedure video of me actually performing a weak D test along with the weak D control. So check that out if you'd like to see it actually in action. So let's talk briefly about Rh antibodies. So most are of the IgG class and react optimally at 37 degrees Celsius and at the AHG phase. So they are stimulated by transfusion or pregnancy, meaning a patient that is Rh negative, meaning they lack the D antigen, can develop an anti-D antibody when exposed to the D antigen via either a transfusion or pregnancy. So Rh antigens are very immunogenic. You do need to know the antigen rankings in terms of immunogenicity. So it's listed here. So little e is the least immunogenic, big D is the most immunogenic, which again, immunogenicity relates to how likely the antigen is to initiating causing an antibody response. So again, very immunogenic, so exposure to less than 1 milliliter of D positive red blood cells can stimulate the production of anti-D in an Rh negative recipient. So I said an Rh negative recipient, so let's think here. Why only Rh negative patients? Can an Rh positive patient develop an anti-D? So no, the answer is no here. So if the patient is positive for any antigen, they do not have the ability to develop an antibody against that specific antigen. And the reason for that is that it has to be a foreign antigen in order for an antibody to form. So the body has to recognize an antigen is foreign in order for the antibody specific for that antigen to be developed. Now, there are times when the body will produce autoantibodies, where the body will just produce antibodies directed at its own red cells, but that isn't what we're talking about here. So if you are Rh positive, you cannot develop an anti-D antibody. Only Rh negative patients can develop the anti-D antibody, and that only occurs if they are exposed to the D antigen through either a transfusion or a pregnancy. Rh antibodies do not bind complement, so this means that it doesn't have the ability to activate the complement system, which is part of the immune system. Rh antigens are not fixed in the red blood cell surface in close proximity for binding complement, so that's why it does not. Most are IgG in class type, and this means they can cross the placenta. So what is the significance of an antibody being able to cross the placenta? So let's say a pregnant mother. Let's say she has an antibody towards a specific antigen, and her fetus happens to have that specific antigen, right? If that antibody is able to cross the placenta, it can attack the fetus's red cells, okay? If it can't cross the placenta, it can attack the fetus's red cells in utero. So Rh antibodies being able to cross the placenta is significant, and it can lead, of course, to hemolytic disease of the fetus and newborn. So I mentioned a couple of slides ago about something called Rh immune globulin. This is also called RhoGAM. So this is a purified preparation of IgG and anti-D. So it's given to D negative women during pregnancy and then also after delivery if the fetus is D positive or Rh positive. So this prevents Rhd hemolytic disease of the newborn and also prevents the mother from developing an anti-D antibody in response to a foreign D antigen from the fetus. Our last slide here describes Rh null syndrome. This is when a patient has no Rh antigens at all. So patients with Rh null can have mild compensated hemolytic anemia with reticulocytosis and stomatocytosis. So in hematology, you'll learn about this. So reticulocytes are an immature form of red blood cells, and stomatocytes are an abnormal red blood cell morphology. Patients with Rh null syndrome can only be transfused with other Rh null blood. Severity of this syndrome depends patient to patient. All right, so that's it for our Rh blood group system. If you have any questions, please feel free to leave them in the comments below. I'm happy to answer any questions you may have. Go ahead and like this video and please share it. And of course, subscribe to my channel for more educational laboratory content. All right, hopefully you enjoyed it and have a great day. Hey fellow lab rats, this is Rebecca from the Lab Rat YouTube channel. In this video, we're going to be discussing the Rh blood system and blood banking. All right, let's get started.